Okay, so what I'm going to do right now is I'm going to go through everything you have to know about cellular respiration. Okay, so it's going to take 10 minutes. You need to watch it, and then if you have questions, you can ask. Otherwise, you'll just do the review. Okay, so in cellular respiration, what happens? You start out with glucose. Okay, and so glucose is going to be converted into what we call pGal. All right, and then finally into pyruvic acid. Okay, and so this step right here, this is glycolysis. Now, to go from pGal, in order for glycolysis to start, 2 ATPs need to be invested, meaning they're going to expend their energy, okay, provide energy to glucose to become pGal, and so they're going to become 2 ADPs. Okay. Now, once pGal has been made, okay, it's going to produce, when it's going from pGal to pyruvic acid, it's going to release energy. So 4 ADPs are going to collect energy from it and become 4 ATPs. And also they're going to release 4 electrons. So you need 2 NAD pluses to grab those electrons, 2 for each NAD plus. They grab the highly reactive electrons because electrons, if they're floating in the cell by themselves, they can be destructive because they're really reactive. So this goes from NAD plus without its electrons to now NADH, because this is an electron carrier. It's carrying electrons. So both ATP and NADH are energy carriers, meaning this created a bond with another phosphate which holds energy, and this one's carrying electrons that are highly reactive. So this is glycolysis. Now, if oxygen is present, meaning it's aerobic, we're going to go through this first, then the following steps happen. You have now the Krebs cycle and the electron transport chain. So the pyruvic acid is going to travel into the matrix. This is the inner pocket of the mitochondria. Okay. And it is what pyruvic acid is, it's a 3-carbon molecule. So 1, 2, 3 carbons. Now, it's going to lose 1 carbon to become a 2-carbon molecule, and it's also going to release 2 electrons. So those 2 electrons have to be picked up by NAD+, which then becomes NADH. Now, I've got a 2-carbon molecule. It's going to bind with coenzyme A to become what we call acetyl-CoA. So this is right at the beginning of the Krebs cycle. Technically, this isn't, but we're just going to call it the Krebs cycle, even though it's not part of the actual cycle yet. Right, now I have acetyl-CoA. When it joins the Krebs cycle, it's going to bind to this 4-carbon molecule, which you guys learned was oxaloacetic acid. You guys don't need to know that. All you need to know is that it's going to bind with this 4-carbon molecule to produce a 6-carbon molecule, which is called citric acid. That's in the fruit that you eat. That acid is what you eat. So this is a 6-carbon citric acid. Now, all that happens in this entire cycle that you're seeing here is this 6-carbon molecule being broken back down into the 4-carbon molecule. So in order to do that, it needs to lose CO2, and now it's a 5-carbon molecule. And I forgot to show this. It's going to lose another CO2 to become a 4-carbon molecule. All right. The other things that happened, it's going to be losing electrons like crazy. So electrons cannot be floating around in the matrix by themselves because they're very reactive, and therefore I need electron carriers to come pick them up. So one of those carriers is NAD+, just like what we talked about before. And so it's going to grab an electron to become NADH. Okay, again, it's going to grab another one to become NADH. And then the other electron carrier that exists is FAD. So FAD is going to go and grab 2 electrons, and it becomes FADH2. Okay, so 2 NAD pluses from right here, and then 1 from right here. All right. Oh, sorry. Yeah, 2, I believe. All right. Then the other thing that's going to be produced is ATP. Sorry. So ADP gets energy from this reaction. Okay, and it stores it in the form of ATP. All right, so the whole point of the Krebs cycle, guys, is to go from that 6-carbon initial molecule and then to become now a 4-carbon molecule, and then it's going to be reused again to make that 6-carbon molecule when it binds to the 2-carbon molecule right there. All right, I forgot, guys, I only wrote 2 NAD pluses. There should be a third. Okay, so NAD plus going in, grabbing electrons again. Okay, notice that it's a 3-carbon molecule. Pyruvic acid is a 3-carbon molecule. For every 1 glucose, there are going to be 2 pyruvic acids, meaning this cycle is going to happen twice. So rather than just 3, remember I forgot this one, 3 NADHs being produced, they're going to be 6 total for each cycle for each 1 molecule of glucose. And instead of just 1 FADH2 being produced, they're going to be 2 FADH2s produced. Instead of 1 ATP being produced, they're going to be 2 for every glucose. So if you're keeping track, glycolysis creates 2 ATPs, okay, for every glucose. Krebs cycle also produces 2. So this is not where most of your ATP is being produced. It's going to happen down here. So ATP, fantastic, we can use it. FADH2 and NADH aren't done yet, okay? Krebs cycle produces them so that they can go down to the electron transport chain, and NADH is going to deliver its electrons, so it goes from being NADH to NAD+, and FADH2 also delivers its electrons to become FAD. So what happens is the electrons are going to move through this electron transport chain, okay, and they're going to be picked up, rather than, if you remember in photosynthesis, they were picked up by NADP+. Instead, since they have less energy because they're slowly but surely losing energy as they move down this chain, they're going to be picked up by oxygen, okay? And we call oxygen our final electron acceptor because it's accepting those electrons. And oxygen with those electrons, oxygen as our final electron acceptor, I keep messing that up, final electron acceptor is going to bind with hydrogen and it makes water. So notice water is being produced in your matrix as a result of the electrons coming off the electron transport chain. The other thing that happens is that as the electrons move through the electron transport chain, hydrogens are being pumped from the matrix into the intermembrane space. This is the intermembrane space. This is the inner mitochondrial membrane, and this is the outer mitochondrial membrane. So there's a whole nother pocket here in between the two membranes called the intermembrane space, and hydrogens are being pumped in here. Now, you're going to get a ton of hydrogen, so a high concentration gradient between here and here. You're also going to have a high positive charge because H+, they have a positive charge. So therefore, what powers the ATP synthase, which is down here, is that movement of hydrogens from here, okay, from that high concentration to here. We call that chemiosmosis because it has a high concentration of hydrogens and also because it's a positive charge. So that's called chemiosmosis. That movement, that tendency for the hydrogens to move from this high concentration area and charged area, intermembrane space, into the matrix. Okay. Now, this ATP synthase is responsible for most of the ATP, or the majority of the ATP that you see in cellular respiration. This ATP synthase, for every 1 glucose, is going to produce about 32 ATPs. So you've only got 2 here being produced, 2 ATPs here being produced, and then 32 to make a total of 36 from this aerobic process. Okay? That is cellular respiration. Cellular respiration in general is aerobic because, except for this one step here, these two require oxygen because oxygen is going to pull the electrons through because it is the final electron acceptor. Without oxygen, this won't function. Without this, right, NADH and FADH2 won't have their electrons removed. Okay, they won't be what we call oxidized. They won't lose their electrons, and therefore they can't be used again to remove the electrons from this. Because if you don't have FAD and you don't have NAD+, your Krebs cycle won't work. So that's why both of these are aerobic, because they need that oxygen to be pulling the electrons through to be getting the FADH2 back to the FAD. Okay, now let's talk about the anaerobic process. So we already talked about glycolysis. Now, when we're talking about fermentation, the first thing that happens is glycolysis. Okay, so notice you're producing pyruvic acid. You're getting 2 net ATPs out of this because you put 2 in and you get 4 out, so net of 2. That's fine. It's not a lot of energy, but it's super fast. Okay, and it can function without oxygen, which is the main point. So here's the problem. This could keep going for an infinite amount of time, except for one fact. This NADH needs to lose its electrons so it can become NAD+ again. And it can't do that unless it's got the electron transport chain, which takes NADH and makes it into NAD+. Therefore, we have two different processes that get my NADH back into being NAD+. The first one, okay, this is the alcoholic fermentation. All it does, it takes pyruvic acid, okay, and it removes, okay, or it provides, the NADH is able to get rid of its electrons to become NAD+. So it's giving its electrons away. So now, thankfully, it's now NAD+. It lost its electrons. So this is the whole point of fermentation, having NADH lose its electrons to become NAD+. So pyruvic acid, once it receives those, 4 electrons from NADH, because it's 2 for each of them, it also is going to lose CO2, and what it produces is ethyl alcohol. So where you see this is in bread, for example. When bread rises, that's a result of the carbon dioxide in the bread, in the dough. Another thing, you see ethyl alcohol all over the place as far as with beers, you see it with wine. The organism that does this with brilliance is yeast. Okay, so in all the alcohols that you can think of, that it's yeast forming that, okay, forming that alcohol. Now, the other type of fermentation is lactic acid fermentation. So the only way it's different from the alcoholic fermentation, it still gets rid of those 4 electrons, right? Because 2 for each NADH, you've got 2 NADHs. It gets rid of 4 electrons, but it does not release CO2. So it produces just lactic acid. And so where you see lactic acid, if you're running a lot, all right, you're going to see lactic acid in your muscles, because in order to get energy as quickly as possible, your muscles do participate in glycolysis, but they still need to get rid of those electrons from the NADH to reuse it, so they participate in lactic acid. Or sorry, they participate in lactic acid fermentation, and so you're getting lactic acid built up in your muscles. The issue with this, the only way to get rid of lactic acid is with oxygen. So once you've sprinted, you're huffing and puffing because your body's trying to get rid of lactic acid, so it's trying to get as much oxygen as possible. That's why you can't sprint for it's a limited amount of time that you can do this, because this will build up. Okay, so after about 90 seconds of sprinting, once you first use up your ATP, hold on one sec, guys. Once you've used up your ATP that is in your muscle cells, and you've done this for 90 seconds, then you have to do this. That's it, guys.